Our red blood cells have tiny markers, or antigens, that act like molecular ID badges. The way scientists test for these markers is almost like a sophisticated matching game. It's called forward grouping, where we mix unknown blood samples with known antibodies to see if they clump together. The ABO blood system is unique because we have naturally occurring antibodies in our blood serum. So while forward grouping looks at what's on our cells, reverse grouping examines what's floating around in our blood serum. These routine methods we rely on today evolved from some pretty dangerous beginnings. In the early 1900s, blood transfusions were basically a game of Russian roulette. Doctors had no standardized way to test blood compatibility. These routine or traditional methods are commonly used method in day-to-day affair in laboratory or blood banking. Newer blood grouping methods offer significant advantages over traditional methods, including increased accuracy, faster results, the ability to identify rare blood group antigens, improved detection of weak reactions, and the potential to test for a wider range of blood group systems, leading to better patient care and transfusion management. In the slide method, we place 4 drops of the unknown blood sample on separate slides and add known anti-sera to each drop. Anti-A to one drop, anti-B to another, anti-D to the third, and normal saline as a control to the fourth. Each mixture is then stirred separately to observe agglutination or clumping. The results can be interpreted as follows. If agglutination is observed with anti-A, the blood group is A. If agglutination occurs with anti-B, the blood group is B. If agglutination is seen with both anti-A and anti-B, the blood group is AB. If no agglutination is observed with either anti-A or anti-B, the blood group is O. Additionally, if agglutination is observed with anti-D, the person is Rh positive. In the tube method, the process or principle is similar to that of the slide method, except it is performed in a tube. It is more sensitive than the slide method. Reverse grouping serves as a recheck system. Here, we follow Landsteiner's law. If anti-A antibodies are found, the person must have type B blood, and vice versa. This method is not used with cord blood or in infants due to the absence of naturally occurring antibodies in their. In this table, we can see the results of forward and reverse grouping, showing agglutination. The Bombay phenotype appears as O group in forward grouping, but shows agglutination with O cells in reverse grouping due to the presence of anti-H substance. In this method, blood and anti-sera are added to microplate wells and observed for agglutination under light manually or with automated analyzers. Gel card method is a newer technique using microcolumns filled with gel or beads to trap agglutinated red blood cells. It provides accurate and reliable cross-matching and antibody screening, widely used in blood banks for its accuracy and reliability. With negative reaction, the red cells pass through the gel upon centrifugation, whereas in positive reaction, the agglutinated red cells are trapped on top of gel or suspended within it. Solid phase red cell adherence assay detects blood group antigens and antibodies using red cell adherence to a solid surface coated with antibodies. It is highly sensitive and one of the commonly used techniques in automated platforms.